Today, a breakthrough designation in multiple myeloma, encouraging findings in 2 HER2-positive breast cancer trials, European regulatory updates in pancreatic cancer and renal cell carcinoma, and an intriguing clinical trial planned in small cell lung cancer. Welcome to Enclave News Network. I'm Gina Columbus. The FDA has granted a breakthrough therapy designation to daratumumab for patients with multiple myeloma following at least one prior therapy. The monoclonal antibody would be indicated for use in combination with lenalidomide and dexamethasone or bortezomib and dexamethasone. The designation, which will expedite the development and review of the daratumumab regimens, is based on findings from 2 phase 3 trials. In the POLLUX trial, the combination of daratumumab with lenalidomide and dexamethasone reduced the risk of disease progression by 63% versus lenalidomide and dexamethasone alone in patients with relapsed refractory disease. In the CASTOR trial, the addition of daratumumab to bortezomib and dexamethasone demonstrated a 61% reduction in the risk of progression or death in relapsed refractory patients with myeloma. In a statement, lead author of the POLLUX trial, Dr. Meletios A. Dimopoulos, National and Kapodistrian University of Athens School of Medicine, said, "Despite tremendous progress in the past 15 years, multiple myeloma remains a highly complex and difficult disease to treat, with most patients relapsing or becoming resistant to therapy. Daratumumab has already shown pronounced activity as a monotherapy in heavily pretreated patients. This designation underscores the potential of daratumumab in combination with either a proteasome inhibitor or an immunomodulatory agent to provide much-needed benefit to patients with at least one prior therapy." The FDA granted an accelerated approval to daratumumab in November 2015 as a monotherapy for patients who have relapsed after more than 3 prior lines, including a proteasome inhibitor and an immunomodulatory agent, or who are double refractory to those therapies. The biosimilar ABP 980 was found to demonstrate similar pathologic complete response rates as the reference product trastuzumab for patients with HER2-positive early breast cancer. The phase 3 randomized study of the biosimilar also had a neoadjuvant stage, in which either ABP 980 or trastuzumab was administered with chemotherapy after surgery. Following this portion of the study, both agents were given as an adjuvant monotherapy. Results showed that ABP 980 demonstrated a PCR rate within a margin of plus or minus 13% compared with trastuzumab, which was the criterion for the equivalence. The upper end of the confidence interval was 13.4%. Additionally, adverse event profiles were similar between the 2 agents, although slightly more events were observed with ABP 980 in the neoadjuvant component of the study versus trastuzumab. In a statement, Dr. Sean Harper, Executive Vice President of Research and Development at Amgen, the company developing ABP 980 in conjunction with Allergan, said, "We believe this study confirms no clinically meaningful differences between ABP 980 and trastuzumab, and we look forward to continued discussions with regulatory authorities. Biosimilars are approved based on the analytical, non-clinical, and clinical data, and we believe that the totality of the evidence we've generated supports ABP 980 as highly similar to the reference product." Biosimilars are meant to foster competition and lower prices, and are defined as biological products that are similar or interchangeable with an FDA-licensed biological product. Also in HER2-positive breast cancer, a 5-year analysis showed that treatment with neratinib reduced the risk of invasive disease recurrence or death by 26% versus placebo as an extended adjuvant therapy for patients with early-stage disease. This was following 12 months of trastuzumab. The analysis of the phase 3 extant study showed that the 5-year invasive disease-free survival rate was 90.4% with neratinib and 87.9% with placebo. In patients who were both HR positive and HER2 positive, the 5-year invasive DFS rate was 91.7% and 86.9% with neratinib and placebo, respectively. Puma, the manufacturer of neratinib, also announced that it had submitted a new drug application to the FDA, including extant data as well as a phase 2 study exploring prophylactic loperamide to prevent the high incidence of diarrhea that is associated with neratinib. The FDA will assign a review timeline for the neratinib application this September. Additionally, an MAA was submitted to the European Medicines Agency for neratinib in late June 2016. Ongoing clinical trials are looking at neratinib in combination with T-DM1 for patients with metastatic breast cancer. A phase 2 study is examining neratinib with or without fulvestrant in patients with HER2 mutant breast cancer. The Committee for Medicinal Products for Human Use has recommended the European approval of pegylated liposomal irinotecan in combination with fluorouracil and leucovorin for patients with metastatic pancreatic adenocarcinoma following progression on a gemcitabine-based therapy. The positive opinion was based on findings from the phase 3 Napoli-1 trial, in which the addition of liposomal irinotecan to 5-FU and leucovorin led to a 1.9-month improvement in overall survival. In the combination arm, the median OS was 6.1 months versus 4.2 months with 5-FU and leucovorin alone. The median progression-free survival was 3.1 and 1.5 months for the combination and control arm, respectively. Additionally, the objective response rate determined by RCC criteria was 16% versus 1%. The CHMP's positive opinion was sent to the European Commission for a final regulatory decision, which is expected within 67 days. Baxalta submitted the application for liposomal irinotecan, which is now owned by Shire. In the United States, the agent is marketed by Merrimack Pharmaceuticals. In a statement, Philip J. Vickers, head of R&D at Shire, said, "This regulatory milestone is an important step for patients with metastatic pancreatic adenocarcinoma who have progressed after gemcitabine-based therapy. There has been little improvement in prognosis for patients in the setting for over 20 years, and given this high unmet need, we look forward to receiving the final approval decision from the European Commission." In renal cell carcinoma, the Committee for Medicinal Products for Human Use granted positive opinions to 2 new therapies for patients with advanced disease. The first agent, cabozantinib, was recommended by the CHMP as a treatment for patients with advanced RCC following failure of VEGF-targeted therapy. This opinion was based on findings from the phase 3 METEOR trial, in which cabozantinib demonstrated a 4.9-month median overall survival benefit when compared with everolimus. Additionally, the risk of disease progression was reduced by 49% with cabozantinib versus everolimus. The second agent, lenvatinib, was also recommended by the CHMP based on phase 2 data that showed a 60% reduction with lenvatinib combined with everolimus versus everolimus alone. In this study, the median progression-free survival was 14.6 months with the combination and 5.5 months with everolimus alone. The European Medicines Agency will make a final decision on both approvals within 67 days. Rovalpatuzumab tezorin is being explored with nivolumab alone or in combination with ipilimumab as a potential treatment for patients with relapsed extensive stage small cell lung cancer in a phase 1-2 study. As single agents, both showed promising results for patients with SCLC in 2 separate studies presented at the 2016 ASCO annual meeting. AbbVie, the developer of Rova-T, and Bristol Myers Squibb, the company marketing nivolumab and ipilimumab, announced the phase 1-2 study in a joint statement. The study is expected to begin patient enrollment in 2016. The trial will focus on the safety and efficacy of the combination. In a statement, Scott J. Dyla, Vice President of Research and Development at AbbVie, said, "We believe the combination of these cancer-fighting agents may offer patients a new treatment option in a disease with limited therapies. By combining immune checkpoint inhibitors that prime the body's immune system to fight cancer cells with Rova-T's approach to target cancer stem cells, we hope to build on our goal to develop differentiated treatments with therapeutic benefit that elevate the standard of care for small cell lung cancer patients." Additionally, the phase 2 single-arm Trinity study was formed to assess Rova-T as a third-line therapy for patients with DLL3 expressing SCLC. 2 phase 3 studies are also assessing nivolumab and ipilimumab as treatments for patients with SCLC. That's all for today. Thank you for watching Enclave News Network. I'm Gina Columbus.